All right, this is going to be the review for mitosis. If you're looking for the review on meiosis, that's going to be the next one we record. So what we're going to be going through is the phases of cell reproduction and really just what is involved in mitosis in general. So this first slide we got here is a picture of a chromosome. Chromosomes are they are made up of DNA, and the DNA is really what is writing the code for proteins. So DNA is what is the formula for proteins, and as the cell gets ready to divide, DNA is in a very long, stringy form called chromatin. Okay, and chromatin is what the structure is before division. Okay, it's a jumbled mess of DNA. And as the cell gets ready to divide, the DNA actually winds up and starts to get into really condensed form. And that's when we get the DNA into chromosomes. So this DNA that is strung out, we actually have 3 meters of DNA in every cell. The DNA that's all strung out is going to wind up around these histone proteins. And you can think of histone proteins like little marbles, or those little like magnetic balls that are attracted to each other. They wind the DNA up around the histones, and then the histone proteins stick together. And as the histone proteins stick together, they wind up the DNA even further until we get into this replicated, replication-ready form, which is the chromosome. The chromosome has a centromere, which you guys can all see here. The centromere is the very center of the chromosome, and on either side of the chromosome is what we call sister chromatids. Okay. These two together are sister chromatids. And what that means is that even though this is just one chromosome, remember to not confuse just the fact that we have 2 sister chromatids stuck together. That doesn't mean we have 2 chromosomes. We still have only 1 chromosome. But we have two sister chromatids here. If we just had a structure that was somewhat like this, saying here's your centromere, this would be 1 chromosome as well. Okay, this is still just 1 chromosome compared to the chromosome that has been written over here. However, this has no sister chromatids because we have no replication of the actual chromosome. So this is still 1 chromosome, but 0 sister chromatids. And that's basically because this form of the chromosome is not replication ready. So there's no reason to have sister chromatids there to divide. So that's really what the center inside that nucleus, really what's happening when the chromosomes are splitting. Before they split, they have to wind up into this form of chromosome right here in the middle, and get ready for that. All right, so this next slide is showing really the cell cycle. What we got here shows the 4 stages that cells go through. We got the M phase, which is actually mitosis, where cells are reproducing or replicating. We have the G1 phase and the G2 phase, where really not much is happening. That's really the normal part of a cell cycle's life. It spent most of its time in G1 and G2. And then we have the S phase, where cells actually replicate that DNA and make an extra copy of it. So all 4 of those phases are in the normal cell cycle. There is one extra phase, it's not written on here, but the G0 phase, which is where cells that have divided already and don't need to reproduce anymore go when there's no cells that are required. So these cells will just sit in the G0 phase, going around and around, and never come out of that phase because they're not required in the body anymore. So as the cell goes through the cell cycle here, there's two things you got to remember. One, that it spends most of its time in these gap phases, and two, that there's certain checkpoints that occur along this cycle in order to prepare the cell for mitosis. So the first checkpoint is right here at G1, the G1 checkpoint, very creative. And in the G1, at the G1 checkpoint, the cell determines whether or not it has to divide. So if there's cells missing and it needs to fill that gap, or there's certain or there's certain requirements that force the cell to divide, it determines that at G1, and it either brings the cell out of the G0 phase, or it just keeps continuing along the normal cell cycle path. So after G1, after it passes that phase, it's going to divide. Okay, it's preparing to divide. As it gets to the S phase, it replicates the DNA. So I'll just write this down here. DNA replication. Okay. And then as it gets through DNA replication, G2 is really preparing that cell to divide, and then there's one last checkpoint right here. Okay, and at that checkpoint, the cell double checks if all of the DNA have been copied correctly, because you don't want to split a cell in half without the DNA accurately being copied. So that phase really prevents the cell from copying bad DNA. And then after that last checkpoint, we get into mitosis, which is those 4 phases. And through the 4 phases of mitosis, the cell really divides and splits into 2. So that's really what's going on here in the cell cycle. All right, so this next picture is really showing the one of the main mechanisms for the division of a cell. As we go through, so we talked about the checkpoints in the last slide. As we go through the cell cycle, this entire time, this phase is called interphase. Okay? And through interphase, the entire time we're going through that cycle, we're building up what we call cyclin. Okay? This level is building up. Now the other enzyme that's present here is CDK. Okay, we can just kind of refer to that as the kinase. And kinases are always present, but cyclin gets built up and broken down as we move through the cell cycle. So as we go through G1, S phase, and G2, this entire time we're going through this, we are building up cyclin. And as we build it up and build it up and build it up, and it gets through to G2 phase, once it gets through that checkpoint, all of that cyclin has been binding with kinases to form the cyclin kinase CDK enzyme, okay, or that complex. So the CDK complex then causes all of that to divide. It causes the cell to actually divide, and then as soon as the cell is done dividing, okay, all of that cyclin is broken down. And then as we go through G1, S, and G2 again, it builds up and builds up and builds up, and then it happens all over. So it's really the cyclin that is determining when the cell divides, because without the cyclin kinase complex, it's not going to divide in the first place. So this is a really important diagram to remember, just so that you know the cell is not just dividing on its own. It's really relying on an enzyme to help it regulate when it divides through the process. All right, and this is really the last diagram I'm going to go through. This is one of the really important ones, and you've probably seen it before. This really diagrams the 4 phases of mitosis. So we got our PMAT over here, and this is something you definitely gotta memorize or find some way to remember. So before we start prophase, before we enter mitosis, we got our parent cell, and the parent cell now has replicated chromosomes. okay, that are going to be splitting up inside the nucleus. So those chromosomes are all wound up into that formation where they have two sister chromatids, and before they enter prophase, they have to be like that. Otherwise, um, the binding microtubules aren't going to be able to pull them apart. So as we enter prophase, the nuclear envelope, as you can see here, starts to disintegrate, and the chromosomes actually start moving towards the center. They're in the process of moving towards the center of the cell, but they're not there immediately. And as they move to the center, those microtubules come across the nuclear membrane and start attaching to the centromeres. And as they attach to the centromeres, they start pushing the chromosomes towards the center and lining them up down the middle. As they get to metaphase, these chromosomes are now lined up the middle, but they're not lined up in their matching pairs. Okay, they're not lined up together. So the homologous chromosomes, they line up randomly. And this is for mitosis. When you watch the video on meiosis, you'll see the comparison between the two processes. But in metaphase for mitosis, all of the chromosomes are just lining up, but it really doesn't matter where they're lining up. So then as we move into anaphase, we have our microtubules pulling the chromosomes apart. So when they're in this X formation, the sister chromatids are attached to each other, and this is one chromosome right here. And now, since we've moved to anaphase, they've actually been split up, and now we have two chromosomes. Even though there was the same X, we've gone from having one chromosome with two sister chromatids to two chromosomes with 0 sister chromatids. So that's another important piece to remember as we go through this cell cycle. And then as we finish and go through to telophase, two things that form are, we have our nuclear envelope forming. So these two nuclear envelopes are new, and those are the daughter cells of the process. And then we have something called a cleavage furrow, which develops in between the two daughter cells. And this process of splitting the two and actually dividing the cytoplasm is called cytokinesis, which is that last phase of cell division. Now, this is what happens in an animal cell, and make sure you remember that this is all for animal cells. Okay? Because in a plant cells can't pinch together and make a cleavage furrow. So instead, what plant cells do is they actually divide the same way using the same processes, but then as they get to telophase, instead of pinching in the middle, a cell plate develops between the two cells, and then they divide in half that way after creating a new nuclear envelope. So through that process, plant cells divide a little differently than animal cells, but instead of having a cleavage furrow, they have a cell plate. All right, so that's really the main points of mitosis, and if you can review PMAT and know the cell cycle and what's going through it, you should be pretty good for the test. So thanks for listening, and we'll see you next time.